video for proteins. Still in biomolecules, we've done carbs, lipids, nucleic acids. Our last one is proteins. There's already pre-formatted note for this in your notebook. It's titled How Do Proteins Keep Us Alive? When you're done typing it up, make sure that you screenshot it and email me a copy, and then make sure you complete that little quiz from today. All right, so once again, we are still in biomolecules. We're doing protein. We've done carbs, which are sugars. We've done fats, which are lipids. We've done nucleic acids, which is DNA, RNA. Also, I didn't mention it in the nucleic acids, but ATP is a type of nucleic acid. It is actually a nucleotide that housed the pool in the basement, but it has 3 pools. So I didn't really talk about that, but I'll talk more about it when we get into cellular respiration. And today we're doing protein, which is basically most things that make you you. So anything that's not sugar, fat, or DNA is going to be a protein. And make sure you know the elements that are involved in them: CHO, CHOMP, and CHON. So in protein we have carbon, hydrogen, oxygen, and nitrogen. Now, you should be familiar with protein because we get it from our food. So some foods that are really high in protein are going to be meat. Like muscle is made primarily of protein, so this chicken breast, well, that's just the muscle on that chicken that helps them fly. It looks like this is some beef, so maybe the leg muscle or the hip muscle of a cow or something like that. There's some fish, and the fish has a lot of muscle. Eggs, that white part of an egg has a lot of protein in it. Also nuts and beans, things called legumes, they have a lot of protein in it too. But hopefully you'll learn that anything you eat, including vegetables, will have at least a little bit of protein in it, because remember, DNA makes RNA, RNA makes protein, and every living thing has DNA. So every living thing has at least some protein. All right, so let's look at the monomer of protein. Remember, monomers are single units or building blocks, and they are called amino acids. Make sure you record it, make sure you know what they are. Here's some chemical structures of amino acids. So every amino acid has this basic structure. There's something over here called an amino group, this N with 2 H's. There's some over here called a carboxyl group, which is a carbon, 2 oxygens, and a hydrogen. And then something really unique about these amino acids is this little side group. It's called an R group or a side chain. And this is going to change depending on what amino acid you have. So every amino acid has this basic structure, but every amino acid, and there's 20 different types, will have a different R group or side chain. Now this slide looks a little confusing, but these are just the 20 different amino acids. Now you don't have to memorize any of them, but if you look at them, like alanine and glutamine and glycine and phenylalanine, maybe you notice that almost all of them end in INE. So ene, alanine, arginine, asparagine. Some of them don't, like aspartic acid doesn't, glutamic acid doesn't, but most of them end in ene. So if for some reason you're ever reading like a STAR test question that has ene on it in this word, it's probably an amino acid, and it's going to be related to protein, right? Alanine is related to protein. So these are the side chains, those different parts of those amino acids. So if you look at these crazy chemical structures, these are the different side chains. And they give these a little bit different structure or chemical properties, which are going to make your proteins kind of look different or be a different shape when they all bond together. Now, speaking of bonding together, it's called a peptide bond. So remember, these amino acids are monomers, and just like all of my biomolecules, these monomers are going to come together to form a polymer. Well, the polymer is going to be called a polypeptide, or a little bit easier way, it's going to be called a protein. So here's one amino acid, here's another amino acid, And you could see in these carboxyl groups and that amino group over here that what I'm going to do is I'm going to combine an H and an OH. So I have two H's, H2, and an O, or H2O, that's water. So if I remove a water molecule here, it's going to bond these two amino acids together. And then another amino acid will come over here. We'll do the same process, pull out of water, they'll bond together. And hopefully you guys remember that this is called dehydration synthesis. So synthesis means to build, right? So I'm going to build this polypeptide, I'm going to build this protein by bonding these different amino acids together. Now, let's look at the actual structure of a protein. So a protein is a polymer, and it's made up of a whole bunch of different monomers, and those monomers are those amino acids. Remember, there's 20 different types. So here's an amino acid, here's an amino acid, there's another one, there's another one, there's another one, and they're going to bond together, and they're going to form a specific shape. When the chemical properties in it will make it twist and turn and bend and form this specific three-dimensional shape. And that's really important to understand, especially when we start talking about enzymes next class. So the shape of a protein is probably the most important aspect of that protein. And every single protein in your body is going to have a slightly different shape, so they're going to do a slightly different job. All right. So the sequence of amino acids, or that order of amino acid, is going to determine that shape. And there's thousands of proteins inside your body, and hopefully you guys remember that DNA makes RNA, and RNA makes this protein. So what a gene is just a section of DNA that's going to code for a specific protein. So every gene is going to have a different order of those nucleotides, of those letters, A, T, C, and G. So every gene is going to have a different order of amino acids, meaning that it's going to have a slightly different shape. All right. And now there are 4 types of shapes for this protein that you need to know. They're the 4 structures of protein. The first one is the primary structure. So this is just like a big long line of amino acids. So each one of these red little circles is an amino acid. So I got, isoleucine, valine, cysteine, glutamine. And if I just keep adding them together, I'm going to make a long line of amino acids. That is the primary structure. It's kind of important to understand that when it's in a primary structure like this, it's just a big long line, and it doesn't have that specific shape it needs, so it's not going to do any type of job inside of your body. It needs to start folding and twisting and turning and bending to get that shape. So when it starts to do that, that is called the secondary structure. So when I start folding it in half, that would be this thing called a beta sheet. Or maybe I start spiraling it, and that would be called an alpha helix. And this big long line of thousands of amino acids is going to start, spiraling and bending, and that's going to be your secondary structure. Now it's going to keep doing that. Maybe these beta sheets and the alpha helix come together to form a bigger structure, and it's just going to keep folding and bending and twisting and turning until it gets this really large tertiary structure. Now this tertiary structure is the protein. This is the shape of the protein, and it's functional. So I have primary structure, a big long line. Secondary structure, it starts twisting or bending over on itself. And then when it's done bending and twisting, that is called a tertiary structure. This is a big 3-dimensional shape, and it does a specific job. Now, sometimes I'll have more than one of these tertiary structures come together, and we'll call that a quaternary structure. So you can see here, here's a protein, here's a protein, here's a protein, here's a protein. So I have 4 proteins together. That would be a quaternary structure of a protein. So primary, line, secondary starts twisting and bending, tertiary is the functional protein, and then quaternary would be an entire protein. Now, I like to equate these to a roller coaster because everybody's been to Six Flags. So the primary structure of a protein would kind of be like the cars here on this roller coaster. So here's one car, it connects to another car, it connects to another car, it connects to another car. And it's just a big long line, right? So that would be my primary structure of a protein. It's the sequence or order of those amino acids. Now when I start spiraling it and folding it back and over each other, that is the secondary structure. And then if I take the entire roller coaster, that would be the tertiary structure. So this is a very functional three-dimensional shape, and it does a job, right? And then if I take like the entire roller coaster park, like every roller coaster that's in there, and put them together, that would be a quaternary structure. So here's one roller coaster, another one, another one, another one. That would be like my entire park. All right, so that's the basic of the structure of proteins. Some of the things that proteins do is like collagen or support. So collagen is one of the things that keeps your skin tight. It's part of the structure of your body. Storage of things like albumin. So I said egg whites have a lot of protein. Well, that's because it's this special albumin. If you're in health science, you'll learn a little bit about albumin too. We give it to people that have a lot of blood loss, helps them increase their blood volume. It transports things like hemoglobin. UHST people, hemoglobin is your red blood cells, and it transports oxygen. Hormones, so some of my hormones are a special type of protein like insulin, the hormone that regulates how much sugar is in your blood. That's a protein. Insulin's related to diabetes. Some of them are receptors, so if you remember right, there's receptors on the outside of my cell membranes, and they help cells communicate with each other. Like my nerve cells, my neurons have a lot of receptors. That's how it sends electrical charges from your brain down your spinal cord, down to the tips of your fingers. Muscles are a big one. So muscle contractions, there's a protein called actin and myosin. That's what makes your muscles contract and relax. So your muscles are made out of a lot of protein. That's why we eat a lot of meat to get protein. Some of your antibodies, part of your immune system, are proteins. And then the big one that we'll talk about next time is going to be enzymes. And what enzymes do is they speed up the reactions in your body, like digestive enzymes will break down your food so that you could actually get it from your intestines into your blood. There's also enzymes that build things up inside your body. And we'll talk more about enzymes in the next class. And that brings us to our central dogma of life, and we said this 100 times. DNA makes RNA, and RNA makes protein, right? So let's look at that process. So remember DNA base pairing, right? So if I have a T, it always pairs with an A. If I have a G, it always pairs with a C. So right here, I have a strand of DNA, TCA, GAG, GTG, AAA, TGC, TAT, GGT. That's my strand of DNA. Well, there's going to be a second strand of that DNA because DNA is double-stranded, right? And they base pair together. So if I have a T right here, the other one will be an A. So I'll put an A here, a C will pair with G, an A will pair with T, G with C, T or A with T, and G to C. And I'll just keep going and going until I have both sides of my DNA. So here's my double-sided DNA. And that brings us to the next part. DNA makes RNA. And hopefully remember that is called transcription. So when I take DNA and I turn it into something called messenger RNA, that is called transcription. So remember this blue part is my DNA. It's a double helix. I have two sides of it, T paired with A, C paired with G. And hopefully remember what happens is my DNA unwinds, and it allows some additional nucleotides to kind of base pair with that DNA. And if I'm going to make RNA, remember there are no T's in RNA. Instead of having a T, I'm going to have a U. So right here I have a T in my DNA. Well, that T is going to pair with an A, so that'll be one of the letters in my RNA. here I have an A, that'll pair with a U, A will pair with a U, G will pair with a C, A to U, G to C, C to G, T to A to U, and I've created here that RNA. And hopefully you guys remember that that RNA is what leaves the nucleus and it goes and finds that ribosome to build a protein. Right? So here's my DNA again. So I'm going to take this template strand, AGT, CTC, CAC, and I'm going to make RNA. And remember, A pairs with U in RNA. There's no T in RNA, it's a U. So A would pair with U, G to C, T to A, C to G, T to A, C to G, so on and so on. And I would create or transcribe this RNA. So A to U, G to C, T to A, so on and so on and so on. Now this is that RNA. This is the messenger RNA that's going to leave the nucleus and it's going to go find that protein or find that ribosome so it can make a protein. So this RNA right here is going to leave the nucleus. It's going to go find a ribosome, which is going to be either on your rough endoplasmic reticulum or floating around in your cytoplasm. And it's going to feed through this ribosome. And as it does, it's going to create a protein. So you see these red little dots. These are all amino acids put together. So this is creating the primary structure of a protein. And the way it does it by using these things called codons. So codons are 3 of my nitrogenous bases. So here I have AUG, that's one codon. ACU, that's another codon. GGC, that's another codon. And one codon, or 3 bases, is going to code for one amino acid. So write this down. A codon is going to code for an amino acid. So it is 3 nitrogenous bases that code for an amino acid. And we're going to use these things, the circle or the square, to figure out which one of those 20 amino acids each codon codes for. So remember, there are 20 amino acids, and these are the amino acids listed on the outside of the circle. These are the amino acids listed on the inside of this big square or rectangle. Now, it looks like there's more than 20 because some of them are on here twice. But we're going to use codons, or 3 bases, to figure out what amino acid it is. And it's actually pretty simple. And these things are called codon charts, by the way. So this is a circle codon chart, and this is a square codon chart. All right, so right here I gave you some RNA, and I know it's RNA because of U in it, right? So GAU, UCA, ACG, this is a RNA sequence. And I'm going to use this circle to figure out what amino acid we're going to make. And it's pretty simple. Let me actually draw it. So we're going to do it with GAU. And in this circle, we're going to start right in the middle with our first letter. So G, we're going to start right here, G. And then our second letter, A, goes in this line right here. So I know my amino acid is right here in this G, but GA, here's my A. So I go G to A. And then my last one is going to be U. So I go G, A, U. And that's going to tell me what my amino acid is. And it's hard for you guys to see, but this is aspartic acid. So it goes G, A, U, aspartic acid. So my first amino acid is going to be called aspartic acid. So I want GAU, aspartic acid. And we'll do it again with UCA. So same way, we're going to start in the middle. So U, here's my U. Then I'm going to go to C, U to C. And then I'm going to go A. So UCA, right there, and it's hard for you guys to see, but it's serine. So my second amino acid is going to be serine, UCA serine. Now the square is Also easy to use. It just takes a couple of more steps. And I'll do it with this UCA again. Now remember, there's 3 of them, right? U, C, and an A. And the square, you always start over here on the left. And it tells you right here, it says first base and the codon. So this is where we start, first base. And our first base is a U. So we go GAC, U. Well, that's where we're going to start, right there, U. So I know that the codon, or the amino acid, has to be in one of these boxes. Now my second one, my second base, is up at the top, and it's a C. So I find the C, and here's my C. And where that U and the C meet, this right here is where my amino acid is going to be. So I have UC, and I already know it's serine, but not all of them are like it. You can see over here, some of them are different. We use the third base. So this says third base in the codon, and our third base is an A. So here's the A. And if I follow it over, I know that that right there is going to be the amino acid I use. So UCA is serine, which is the same one we got here. So I have U, I have C, and A, and where they all come together, the amino acid is serine. Let's do the box with ACG. So we're going to start over here again, A. So here's my A. Then I'm going to find my C, second base. So C, also right here. So where A and C meet, I know the amino acid is going to be right in here because A and C meet right there. And then the last letter is going to be G. So we find the G over here. Here's my G. And where it comes over, it's going to be threonine. So ACG is threonine. And we look over here, ACG, it is also threonine. So they get you the same answer, and you're going to need to know how to use both. These are going to be on your STAR test. We're not sure which one, so make sure you know how to use both of those to come up with the sequence of amino acids. All right, now there are a couple special codons. One of them is called a start codon, and it simply is the start of your protein. It's kind of interesting because every gene that we know of in every living thing starts with methionine, or this codon of AUG. So I have an A, I have a U, I have a G. Methionine is always the very first codon for every protein that's made, or methionine is the first amino acid for every protein that's made. There's also some stop codons, and those simply mean that it's telling the ribosome that the protein is done being made. So when a stop sequence comes up, the ribosome knows, okay, we're done, and it releases that primary structure of the protein. And you can see there's 3 of them in here. So I have a stop codon, a stop codon, a stop codon. If I look at the sequence, the stop codon, this one would be U, A, that's one stop codon, or U, A, G, that's another stop codon. And then my third one is going to be U, G, A. So if you have any of those sequences, those codons, it tells your ribosome that you're done making the protein. All right, so let's see this whole thing in action. Remember, we have our messenger RNA, UCA. We'll use a codon chart to figure out what that amino acid is. So UCA makes serine. So here's my first amino acid. GAG makes glutamic acid. There's my second one. GUG, so GUG, valine, that's my third one, or third amino acid. Then I have AAA, lysine. Lysine, that's my fourth amino acid, and so on and so on and so on. Now, when you guys do this, sometimes they'll give you the RNA sequence, and it's really easy. You just use that codon chart. But sometimes they'll give you the DNA sequence instead. And you'll be able to figure it out just by looking at it. If there's a U in it, it's going to be RNA. If there's a T in it, it's DNA, and you're going to have to turn it into RNA first before you can figure out what these amino acids are, because there are no T's in here, right? So like the DNA sequence here would be A, G, T. So if you had an AGT here, you would have to turn it into UCA first to figure out what that amino acid is. And here's my sequence of amino acids. So serine, glutamine, valine, lysine, so on and so on. This is that primary structure of that protein. And then it's going to start twisting on itself. It's going to start forming those alpha helix and beta sheets. And then it's going to keep on turning and twisting and moving until it has that tertiary structure of a protein. And then it's going to possibly turn into a quaternary protein. And that will be a big functional protein. Right, so that is the basics of protein and transcription and translation. Hopefully you guys understand it. We will be doing a lot more using these codon charts coming up, so you'll get some more practice turning DNA into RNA and RNA into that primary structure or that amino acid sequence. So make sure you type up your notes, take a screenshot, email it to me. If it's complete and on time, you'll get 100. If it's late, I'll start taking off points. All right, thanks.